Look closely at this cellular surface reacting to a stimulus. At the microscopic level, inflammation functions as a highly deterministic, stepwise algorithm. To execute a defense response, the body runs two parallel biochemical engines simultaneously. The first is the cell-derived engine. This relies on chemical agents synthesized directly on site by local immune cells, or immediately released from internal storage granules the moment tissue is damaged. The second is the plasma-derived engine. This utilizes a cascading sequence of pre-existing proteins that constantly circulate in the bloodstream, waiting for an activation signal. Today, we're going to map the two most critical pathways governing this dual system: the cell-derived arachidonic acid cascade and the plasma-derived complement system. Mapping this architecture is the absolute prerequisite for mastering clinical pharmacology and immunopathology. You cannot treat what you cannot trace. This vector model represents a standard cellular membrane's phospholipid bilayer. The fuel for the cell-derived engine begins as a physical structural component of the cell itself. When a cell is damaged, an enzyme called phospholipase A2 shears these phospholipids apart. This creates an isolated fatty acid node called arachidonic acid, or AA. Arachidonic acid is the raw material. From here, it routes into two competing enzymatic pathways: cyclooxygenase, or COX, on the left, and lipooxygenase, or LOX, on the right. Let's follow the COX branch first. The COX enzymes convert arachidonic acid into an intermediate molecule called prostaglandin G2, which rapidly becomes prostaglandin H2. Prostaglandin H2 acts as a central hub. One specific output is prostaglandin E2. This molecule sensitizes local nerve endings to induce pain and signals the brain to raise the body's temperature, causing fever. Next is thromboxane A2. This acts on local blood vessels to cause vasoconstriction and facilitates platelet aggregation, initiating clot formation. To prevent unchecked clotting, the same pathway produces prostacyclin, or PGI2. This is the regulatory counterforce, causing vasodilation and inhibiting platelet aggregation. Now let's trace the other half of the tree. The lipoxygenase pathway uses the enzyme 5-LOX to convert arachidonic acid into a family of molecules called leukotrienes. Leukotriene B4 acts as a potent chemoattractant. It serves as a chemical beacon, recruiting white blood cells, specifically neutrophils, directly from the bloodstream to the site of the injury. The remaining nodes, LTC4, LDT4, and LTE4, act directly on smooth muscle as potent bronchoconstrictors, severely narrowing the airways. Knowing this geometry allows us to manipulate the entire response. Corticosteroids inhibit phospholipase A2. This shuts off the raw material supply and halts the entire cascade. Non-steroidal anti-inflammatory drugs, or NSAIDs, act more selectively. They block the COX enzymes, eliminating pain and fever mediators on the left, while leaving the leukotriene pathway on the right intact. By mapping this exact enzymatic geometry, identical source molecules can be chemically manipulated to produce or halt contradictory physiological responses. Immune cells are releasing bursts of glowing particles called cytokines. While lipid pathways dictate local vascular changes, cytokines dictate the systemic physiological state. Specific interleukins drive different parts of the systemic reaction. Interleukin-1, interleukin-6, and tumor necrosis factor alpha act as pyrogens, traveling to the hypothalamus to induce fever. Other cytokines act strictly as movement instructors. Interleukin-8, for example, is a CXC chemokine, whose sole purpose is the recruitment of neutrophils into the tissue. The system also requires a regulatory brake. Once the pathogen is cleared, interleukin-10 acts as the primary anti-inflammatory cytokine to initiate tissue resolution. If arachidonic rhythid mediators are the local hardware changes of inflammation, cytokines are the software instructions broadcasting priority alerts to the entire body. Now let's examine the plasma-derived engine, the complement system. This flowchart tracks a sequence of proteins, labeled C1 through C9, that float inactive in your blood plasma, waiting for a target pathogen. The classical pathway is triggered when an antibody, specifically IgG or IgM, binds to an antigen on the pathogen surface. This mechanical link activates the first protein, C1. Once activated, C1 cleaves proteins C2 and C4, combining their fragments to construct the classical C3 convertase. The system has a built-in redundancy called the alternate pathway. It is triggered by IgA antibodies or direct physical contact with microbial surface molecules. This alternate route utilizes a circulating protein called factor B to build the alternate C3 convertase. Notice the structural convergence here. Despite relying on different recognition triggers, both pathways exist solely to cleave the C3 protein into two fragments, C3a and C3b. The C3b fragment is a potent opsinin. It physically coats the surface of the bacteria, flagging the cell for destruction by macrophages. The plasma engine's architecture is engineered to funnel every recognition trigger toward a singular, overwhelming amplification loop at C3. The C3b protein then binds back to the existing convertase complex. This physically alters the enzyme to form the next stage, C5 convertase. This new enzyme complex targets the C5 protein, cleaving it into fragments C5a and C5b. Much like LTB4 in the cellular engine, C5a is released as a powerful chemotactic agent to recruit more immune cells, but C5b is the fragment that initiates the terminal assembly. C5b recruits proteins C6, C7, and C8, followed by a repeating ring of C9 units. They self-assemble into a rigid structure called the membrane attack complex, or MAC. This simulation shows the physical execution. The membrane attack complex acts as a microscopic drill, driving a protein tube directly through the bacterial lipid bilayer. By forcing a physical opening in the membrane, osmotic pressure drives water inside the pathogen. The bacteria swells and undergoes cell lysis. The membrane attack complex is the terminal mechanical output of the complement engine. It is the specific point where a recognition cascade translates into direct pathogenic destruction. When we place these systems side by side, the full picture emerges. Acute inflammation requires this exact dual machinery to function correctly. There is a clear division of labor. Arachidonic acid controls vascular access and cellular recruitment, while the complement system executes the destruction of the pathogen. A genetic deficiency preventing the formation of the membrane attack complex leaves the body without its terminal drill. This results in high vulnerability to recurrent infections from Neisseria species. Conversely, a deficiency in the C1 inhibitor allows the complement cascade to fire unchecked, resulting in severe systemic angioedema. At its core, inflammation functions as a highly targetable, stepwise biochemical algorithm. By mastering these biochemical flowcharts, medical professionals stop reacting to surface-level symptoms and gain the ability to precisely rewrite the body's defensive software.